Okay, we're still in Unit 1, Scientific Method and Microscope, and this is Lesson 2. And we're going to continue talking about the scientific method. So let's make sure you're on the right page, Experimental Design Matrix, Choice 1. And we're going to review the different parts of the scientific method using an example. So remember, the title of the experiment tells us what we're trying to find out. It's a statement of what's being investigated, and it includes the independent variable, the dependent variable, and what's being studied, or the species. And it's usually written in the form of the effect of the independent variable, that's what you're changing, on the dependent variable in some species. So usually this starts with a question. So I'm going to write a question over here. So the question that I wrote is, will giving tomato plants nutrient food increase the size of the fruit produced? So that's our question. So when we look at this, our independent variable is going to be nutrient food, because that's what we're going to be changing. That's going to be our independent variable. And the size of the fruit is going to be our dependent variable. And we're using tomato plants. And we're going to pick a type of plant because there's many different species of tomato plants. So it'll be the effect of nutrient food on the size of fruit produced. So tomatoes. And we'll pick a species of tomato plants. We'll say Roma tomato plants. Those are the plum tomato plants, the ones that they use, the plum tomatoes that they use like in Italian sauce. Okay, so there is our title. Next, let's move on to the hypothesis. What do you predict will happen during the experiment? So what's your educated guess? And this again is usually in the form of if-then. If I do this, then this will happen. So what do you think will happen if we give tomato plants nutrient food? Well, hopefully you said that you think that they'll get bigger. So if Roma tomato plants are given nutrient food, then, there's the then part, then the size of the tomatoes produced will increase. So that's our educated guess about what we think will happen. Okay, what is our independent variable? So what are we changing? Well, we said the independent variable is the nutrient food. We're going to change the amount of nutrient food. So levels of the independent variable. So we might have 0 milliliters, 10 milliliters, this is of the nutrient food, 20 milliliters, we'll double it, and 40 milliliters. And how many times? We have to do repeated trials. So let's just repeat each of these 3 times. We'll have 3 different plants. So we'll have a total of 12 plants, and we'll do 3 at 0, 3 at 10, 3 at 20, and 3 at 40. So what is the dependent variable? What are we measuring? What data are we going to collect? Well, we can measure the mass in grams, so we'll put G, of tomatoes. And maybe we want to measure the circumference. Measure the circumference in centimeters of tomatoes. So that's the data that we would collect. Okay, what's our control variable? Those things that you need to keep the same during the experiment so that you are only testing your independent variable. Well, we need to keep the same amount of light. We need to have them all in the same soil. We need to have them all at the same temperature. And they all need to be the same type of plant. So our control group gets none, or the normal amount of what's being tested. So the control group gets 0 milliliters of nutrient food. So another thing that they all have to get water. So they're all going to get water, and they'll all get 20 mils per day of water. Because they have to have some water, they won't grow at all. And then group 1 can have 10 mils of nutrient, 20 mils, and this one will be 40 mils. And that's where we are going to stop for today's lesson. Okay, see you in class.